Hello, I'm Randy Bateman, and I'll be presenting the autosomal dominant Alzheimer's disease, primary and secondary prevention of dominantly inherited AD for the Dominantly Inherited Alzheimer's Network Trials Unit. As part of this presentation, I'll be reviewing some of the background and rationale for prevention trials in Alzheimer's disease in general, as well as the specific rationale for dominantly inherited Alzheimer's disease. The disease has been known for some time in its form of a familially inherited disease that affects people at an early age of onset. In fact, the first patient of Dr. Alzheimer that he described potentially had a mutation in her presenilin-1 gene. August Dieter also had a potential family history and an early onset in her early 50s. The mutations were discovered many decades later in the 1990s in amyloid precursor protein, presenilin-1, and presenilin-2. It was learned that mutations in these three genes all appear to change the metabolism of amyloid beta, either making stickier forms or making more forms of the longer 42 isoform of amyloid beta. These invariably lead to disease in these people, and as a dominantly inherited mutation, are passed down through the generations at a 50% chance for each child to get it. And importantly, there is a highly predictable age of onset based on the family and the mutation type within the family. The dominantly inherited Alzheimer's disease is similar in many respects to sporadic Alzheimer's disease, but there are some important and notable differences. They clinically present with an amnestic phenotype, memory loss, typically short-term memory, progressing to long-term memory and other cognitive disabilities, and are associated with a characteristic atrophy seen in the hippocampal regions and the cortex. The amyloid deposition appears to start in the higher association cortex regions, and in some families starts in the basal ganglia as well, as measured by amyloid PET scans such as PIB-PET. Some years later, this is associated with hypometabolism in the parieto-occipital regions, and the characteristic biochemical changes in cerebrospinal fluid show a drop in the amyloid beta 42 amounts and an elevation in cerebrospinal fluid tau and phospho-tau species. To better understand this population, a research network was established in 2008 called the Dominantly Inherited Alzheimer's Network, or DIAN, and a few years later, the trials began with Dominantly Inherited Alzheimer's Network Trials Unit, or DIAN-2. Shown here are the trial sites. The current ones are shown in red, and the sites that are in the process of joining the DIAN network are shown in blue. And so you'll see that there's a broad representation globally of these centers. Through this work, the network has established a sequence of changes starting in the left in the top panels. You can see the changes of amyloid deposition and growth throughout the cortex, and as well as the basal ganglia. And that's followed at about 7 years before symptom onset with hypometabolism in those characteristic regions. At about 5 years before, the cortical atrophy begins setting in and progressively gets worse once we pass symptom onset at 0 into the symptomatic years. The atrophy and the hypometabolism continue to progress. I'll let this video play through one more time just to point out that the sequence of changes that occur in the brain are informative in terms of which processes may lead to downstream effects. So based on this and the genetic evidence of dominantly inherited Alzheimer's disease, we believe that amyloid deposition and aggregation are one of the earliest events, followed by hypometabolism and at symptom onset, here at the estimated years to onset of 0, atrophy of the brain. And as we look in other newer modalities, such as tracking tau aggregation with tau PET scans for tangles, you can see the similarities between sporadic Alzheimer's disease on the left and familial or dominantly inherited Alzheimer's disease on the right. And what you can see is that even though the regions are characteristic and similar in the temporal lobe, the parietal lobe, the occipital lobe, and some in the frontal lobe, the amount of tau pathology is greater in that of the younger onset autosomal dominant Alzheimer's disease on the right. And so this suggests that even though the disease is the same disease and the processes are similar, that there are some unique characteristics in this younger onset population. Quantitatively, we can graph this by the numbers of years until expected symptom onset, and that's defined here as 0 based on that particular mutation carrier's average age of onset. And we can look 10, 20, even 30 years before symptom onset and up to about 10 years after. Most people pass away from this disease in 10 to 11 years after symptom onset. And what you'll see is characteristic changes, again, of amyloid beta increasing, shown here in orange, and subsequently followed by changes in cerebrospinal fluid. In this case, total tau. We now know quite a bit more about the changes in tau that occur in these leading stages up to symptom onset with the clinical dementia rating, sum of boxes increasing to significant levels at 0 on average. And these stages, now that we have the tau PET, we also know that tau PET really begins very close to symptom onset. And so the aggregation of amyloid and tau are separated by about 20 years, and there's soluble tau changes that occur in the intervening period. We can define these stages based on when the pathology occurs. So tangles predominantly occur in the symptomatic stage in this population, while amyloid plaques largely occur 15 to 20 years before symptom onset and reach near maximal levels after symptom onset. And then we define the stage before that, before plaque deposition, as pre-pathology, before there's detectable Alzheimer's pathology in the brain. And so based on this, the DIAN-2 trial platform design launched initially with a key aim of testing diverse mechanisms of action in amyloid beta, thought to be the initiating event in this population. And as we set out to do that, we launched with 2 different monoclonal antibodies and also launched a beta secretase inhibitor to lower the production of amyloid beta. We are planning now to launch drugs that target tau, and we hope in the near future something with combination treatments. One of the goals of the platform is to test Alzheimer's disease at the different stages, pre-symptomatic as well as symptomatic, in what we call secondary prevention. And now we have a trial plan for primary prevention before the amyloid plaques form. We plan to launch a study for that. The platform uses a pooled placebo design. That pooled placebo design is there to increase the power to detect a drug effect and also to maximize the potential impact of drugs on individuals. The platform is designed to be adaptive, and so when new findings from the field come in, the trial can change individual arms, doses within those arms. We can add biomarkers with the growing biobank and dataset platform that we have, and the trial continues to grow in its power as this data is collected. In order to launch a trial like this, it took an enormous number of groups and individuals to bring this to fruition. And so shown here are the many groups that really helped start and establish and continue to support the DIAN-2 platform, including the Alzheimer's Association, other philanthropic groups such as GHR and foundations. And importantly, the National Institute on Aging has supported all of the trials, making the data and the samples publicly available. Our pharma partners who sponsor their drugs and their programs within the trial are key and essential members, and a pharma consortium helps inform next steps and advice on the platform as it evolves. So now I'm going to review the findings from the first 2 drug arms, from solanezumab and gantenerumab. And we launched this at the end of 2012 as a double-blind, placebo-controlled trial, initially a biomarker trial that was then changed from a 2-year to a 4-year cognitive endpoint trial with a biomarker interim analysis. The population is the secondary prevention population and early symptomatic population. So people who have a mutation or are at risk of having the mutation and are between 15 years before their symptom onset and no later than 10 years after symptom onset, while still having very mild or mild dementia, so CDR 0.5 or 1, or CDR 0 for the asymptomatic people. It was a pooled placebo design, as I mentioned. We initially planned to enroll 34 placebo individuals and then have 52 actives for SOLA and 52 actives for GANT. And we'd also planned that there would be people who don't have the mutation. They would automatically be assigned to the placebo, and they're in the trial because most individuals don't know what their mutation status is, and most did not want to know what their mutation status was. So the trial was designed to enable people to participate without forcing them to know their individual genetic status. As I mentioned, we had a 4-year trial design, but this was a common closed design, so we expected to collect data after the 4 years. And the overall summary for GANT and SOLA are shown here. We had developed a novel cognitive endpoint and a Bayesian analysis based on that called the multivariate cognitive endpoint. And in that primary endpoint, neither drug showed benefit. for a cognitive slowing of the disease. This is confounded, and the interpretation of whether there could be a cognitive effect is limited due to several factors. One of those is that we increase the dose to what we think are more efficacious doses later in the trial, and I'll show you some of that. That it was really 2 to 3 years after the trial had launched before we were able to increase those doses. In addition, the symptomatic group who was on the low dose, they had declined significantly before they were able to go on the high dose. And conversely, the asymptomatic group had very little decline, or in fact, in overall improved during the trial, but improved for all groups, the active treatment groups and the placebo group. So we weren't able to determine a treatment effect in the asymptomatic population. No new safety signals emerged. Both drugs engaged their targets. Gantenerumab reduced brain amyloid levels, and solanezumab bound significant amounts of soluble amyloid beta. In addition to that, gantenerumab demonstrated highly significant effects on downstream measures of Alzheimer's disease pathophysiology, including lowering cerebrospinal fluid total tau, cerebrospinal fluid phospho-tau 181, and improving the amyloid beta 42 to 40 ratio towards normal. It also had a limited effect in slowing the increase of neurofilament light chain, thought to be a measure of neurodegeneration. So here's the data, and gantenerumab's target engagement is shown here on the left. You can see that gantenerumab had significantly decreased the amount of amyloid plaques as measured by PIB PET scan and compared to placebo, and that this was a relative reduction of 200%, and the difference was around 26 centiloids. So highly significant decrease. When we took a look at this by stage of disease, so those who were symptomatic on the left, clinical dementia rating greater than 0, versus CDR 0, asymptomatic on the right, you of course see the higher amounts of amyloid starting for the symptomatic versus the asymptomatic group, but you also see some differences. And what we noticed was that by year 4, we still hadn't gotten people to the normal range yet, and that it would either take more time or higher doses to do that. Some of the individuals in the asymptomatic group may have been improving, and for comparison, if someone did not have a mutation in the trial, they were very close to 0 in their amyloid deposition. On this slide is shown the gantenerumab's effects on several different biochemical measures in the cerebrospinal fluid, and one of the things that was impressive to us was that while the placebo group continued to increase in the total tau and the phospho-tau 181, gantenerumab clearly decreased those levels towards normal, normalizing those values by almost a third, 30 and 32%, towards a normal total tau and phospho-tau 181 level. Similarly, for CSF beta 42, gantenerumab raised those levels towards a normal ratio, while those who were on placebo didn't really change much. They continued to slightly trend towards abnormal, and a 40%, 42% difference there, normalizing that ratio. And the CSF NFL, you can see here, placebo had gone up some during the trial, while the gantenerumab did not really increase so much during the trial. So based on this biological evidence, and based on the fact that we couldn't draw a conclusion on the cognition, we and our partner Roche have decided to have an exploratory open label extension. And this is based on the biological effects, the acceptable safety profile, and that further study is warranted, and that we want to answer more questions related to this promising drug. So what are the questions that remain? One is, can gantenerumab completely remove amyloid plaques in this population of dominantly inherited Alzheimer's disease? And can we move that to normal levels? Can we get it below the threshold of abnormality, which is around 20, or even towards 0 by a centiloid scale? If we continue to reverse those amyloid plaques, can we normalize other Alzheimer's disease downstream measures, such as soluble and aggregated tau? We have soluble measures. We've instituted tau PET within the trial partway through when it became available, and we'd like to study that to understand whether the tau aggregation is also affected. And how much can we normalize it or slow down the growth of tau tangles, which we think are very important clinically and cognitively. And furthermore, if we continue treatment and decrease amyloid plaques, can we have a larger effect on neurodegeneration markers, such as the neurofilament light chain I mentioned, or imaging measures? And importantly, do these correlate with changes in cognitive or clinical outcomes? If we follow people long enough, eventually the asymptomatic people will or won't have cognitive and clinical decline. And we think that that'll be an important measure to at least begin to assess. And ultimately, is removing plaques a valid strategy for prevention in dominantly inherited Alzheimer's disease, but also sporadic Alzheimer's disease? So based on those questions, we wanted to offer this to the participants who had been in the trial, 4 to 7 years for some, and who had been on placebo, and some had been on solanezumab and others on gantenerumab. And so this open label extension will give them that opportunity to continue to participate. And we want to inform of completely removing the amyloid, as I mentioned on the prior questions. And finally, how will this inform us in our next steps, our next trial designs? I want to finish by telling you where we're going at in the future, and what trials are now in trial arms in the platform are coming forward. Tau is incredibly important in Alzheimer's disease. It appears to be a strong effector of the cognitive and the clinical manifestations of the disease. And so based on that, we've been focusing extensively in this population. Shown in this graph is a publication from early 2020 from Nicholas Barthelemy and Eric McDade, looking at novel tau measures. And you can see phospho-tau 217, 181, and 205, that these appeared to occur in a sequence of changes with total tau later, before the tangles form, the neurofibrillary tangles form here in red. And so this sequence of changes, we think, can be quite powerful to inform on tau measures, in addition to the tau PET, which we're now collecting. And so our plans are to launch several tau-based drug arms in the platform, and to begin to consider, can we combine tau drugs with amyloid drugs in a combination therapeutic trial that may have the best chance of success. And finally, what we and others, some others, consider maybe an ultimate trial is, can we do primary prevention? Can we really prevent even the beginnings of the pathology, the amyloid plaques, from forming in the first place? And we know that that is a very early time for these individuals, and probably sporadic AD as well, but it does offer the best chance of having an absolute success. And the prediction here is, if we can really prevent the pathology, that these people shouldn't develop dementia. But this is a very challenging trial. It's being led by Eric McDade within the DIAN-2 platform, and that this prevention will need to be started at the earliest time. So people as young as 18 years old, 18 in their early 20s to early 30s, will be enrolling in this trial and be taking interventions designed to prevent amyloid plaque formation, and to then determine, are these downstream biomarkers affected, such as cerebrospinal fluid tau, and eventually tau tangles, and does cognitive and clinical measures, are they improved? So here's a roadmap of where we're at. We're currently 2020. We're now in the open label extension of gantenerumab. We had launched and stopped the beta secretase inhibitor arm, and the cognitive run-in for the next trial arms is active, and people have enrolled and are being followed in this cognitive run-in period. We're preparing to launch some of our next tau next-gen drug arms, as well as the primary prevention trial in 2020 to early 2021. So I'm going to end thanking everybody and pointing out we have resources. If family members, researchers, or others would like information on these studies, they can contact us at these links, emails, and phone numbers. And to recognize the incredible work of all the DIAN centers shown here, and the leadership within DIAN. Here's a photo of our steering committee, as well as the DIAN-2 groups that help lead these trials. Shown here, the many people who have been instrumental, and there are many more that we can't list on the slides, who have worked over the past 7 to 10 years to get these trials to be a success. And this includes our centers in many areas led by leading medical research groups, as well as future sites. Thank you.